Day 237 of isolation. Things are getting dark. I've expended all of my razors, and I'll have to resort to shaving with a Bowie knife, but I've yet to be brave enough to try that. Food sources depleted, and it's too dangerous to go to Wegmans. I was forced to eat my rabbit, Fluffy. The worst part, he was delicious. I don't know if I have the heart to go on. And there's my segue. Hi guys, welcome back. Today we're going to be talking all about the heart and the cardiovascular system. Ignore that little stunt at the beginning there, hopefully to lighten your mood after this social distancing conundrum that's bound to go on for a bit longer. So today we're talking about the cardiovascular system. Last week we talked about the nervous system, and these are the two units which will be part of Practical 3, which you have next week. So before we get too far into things, I want to go over some of those prefixes and suffixes on your list. So the first is artery. I'm sure you've heard of arteries before. Their main purpose is to carry blood away from the heart and towards your body or towards your lungs. So arteries go away from the heart, and in general they carry oxygenated blood. Vein is the opposite. Veins come back towards the heart, and in general they carry deoxygenated blood, which needs to pick up some more oxygen from the lungs. Cardi or cardio refers to not Cardi B, but instead the heart, anything pertaining to the heart. And erythro is a long suffix which just means red. So we go on to leuco, which means white. So we have red blood cells and white blood cells. Their formal names are erythrocyte and leukocyte. Which brings us to the one I skipped over, cyte. Cyte is a type of cell. We have chondrocytes, those which maintain cartilage. We have erythrocytes, red blood cells, leukocytes, white blood cells, pneumocytes, cells of the lungs. Pulmo is another suffix which can mean pertaining to the lungs. And we have papilla, which is a rounded protuberance, which means sort of a bump. But in this case, the bump, instead of being of bone, is going to be of tissue, in this case muscle. We'll get to papillary muscles a little bit in this presentation. And then vas refers to a duct or a structure. So the vasculature of the body refers to the arteries and the veins, ducts. So the cardiovascular system is including the heart, the blood, and the blood vessels. And its main purpose is to move things around. It is sort of the public transit of the body. We are talking about moving wastes such as carbon dioxide, urea, salts we don't need, as well as moving nutrients being oxygen, as well as glucose or fats or things that our cells need like amino acids. So these are moving around in the blood, and the blood is our means of moving them to where they need to be. So we need to talk about what blood really is. You see that bloody handprint up in the corner, and hopefully when we talk about blood, we're referring to anatomy, not some blood that has been lost by somebody. Blood is a connective tissue, believe it or not. We call it a connective tissue even though it's a fluid. And the first part of your packet this week is talking about what's in blood. So we're going to spend some time talking about the composition of blood. So, and we already talked about its function to carry nutrients and wastes around the body from where they start to where they need to go. So the composition is really the big question we're asking here. So most of blood, 55% of blood, is plasma, and we'll break down plasma also. And then we have formed elements like platelets and white blood cells and red blood cells. So the formed elements are red blood cells, white blood cells, platelets. And we'll go on. So plasma. Plasma is sort of the watery goo that all of the cells are suspended in. So plasma is the non-cellular or cell-related material. Plasma is mostly water. It is 92% water. It is about 7% proteins and about 1% solutes. Solutes being calcium, sodium, chloride, glucose, amino acids, anything floating around in the blood that's dissolved in the blood, as well as the gases like oxygen or carbon dioxide, and hopefully not carbon monoxide. Next, we have erythrocytes, which are by far the largest formed element. Not largest in size, but largest in abundance. So we have erythrocytes, the red blood cells, at 45%. In your packet, it says it's 99.9%. That is 99.9% of the formed elements total. And then we have less than 1% leukocytes and less than 1% platelets. So if you were to take a blood sample and put it on a microscopic slide, or a microscope slide rather, and look at it, you would see mostly red blood cells. You would see very, very few leukocytes, and you probably cannot see the platelets. So let's get into blood plasma. Blood plasma is the most abundant factor of blood. It is mostly water. We already talked about that it's proteins and solutes dissolved within. And then something we need to talk about is osmotic pressure. That's something your packet mentions. Osmotic pressure is the idea that water wants to flow in a certain direction. So when you have salt dissolved in water, like the case is for the cytoplasm of cells as well as for the blood, the concentration of those salts and the concentration of water wants to be roughly equal. So let's say you take in a lot of salt and there's a lot of salt that's in your blood plasma. Well, now your cells want to drain out some of their water to raise and equalize the concentrations of salt inside and increase the concentration of water outside. So osmotic pressure is this kind of complex idea that unless you take another biology course, you're not going to spend too much time talking about. But osmotic pressure has to do with why when you eat a lot of salt, people feel bloated and swollen, because water is moving out of their cells and into their blood vessels. And so there's some movement of water there. Osmotic pressure refers to water movement. So formed elements, red blood cells. By the way, 2 red blood cells fell in love, but it was all in vain. Sorry about that. I have to tell some jokes to get through isolation. Red blood cells, their other name erythrocytes. Erythro, remember, means red. They are anucleate, which means that they do not contain a nucleus. That's kind of weird, a cell without a nucleus. And we have to think that at one point they did have a nucleus. So when they were developing cells, they had a nucleus, and then they ejected it. They got rid of it. We don't want this. We don't need this. The reason they don't have nuclei or mitochondria is because red blood cells are meant to carry oxygen. Oxygen is a molecule that we need to produce energy. So if we had mitochondria and a nucleus in our red blood cells, that would mean those cells themselves need to use the oxygen to make their energy. So imagine you're starving and you're going to the donut shop because your friends asked you to. And they say, by the way, don't eat any donuts on the way back. Well, that's not going to happen. That's sort of the instance that we're talking about here. These cells have expelled their organelles which use energy so that they can transport these molecules without consuming them to make their own energy. They get their energy in another cycle. So they transport oxygen and carbon dioxide. They take oxygen, which is picked up in the lungs, the alveoli of the lungs, and they bring it to all of the tissues of the body where it's deposited and then used by the cells. Through cellular respiration, we produce carbon dioxide. Carbon dioxide needs to leave the body, so it will follow red blood cells out to the lungs where it's expelled, and then the cycle can continue. In particular, red blood cells contain a protein you've probably heard of before, hemoglobin. Anytime you hear that word hemo or hema, think of blood, relating to the blood. Hemoglobin is the protein of interest which carries the oxygen molecules. It contains an iron 3+ or an iron in its +3 oxidation state, and that's what gives blood its characteristic red color. So I have 2 pictures here for you. Remember, you can follow my cursor. The top picture is sort of an animation of a red blood cell. It's a biconcave disc, which basically means it kind of looks like a donut, except there's not a hole in the center. It's just a depression. And this increases its surface area, so more oxygen can bind all the way around these cells. What we're looking at here is a stain. So we took a blood sample on a microscope slide and we stained the cells, and this is what we get. So you'll see that we can see tons of red blood cells because they're super abundant. And right here we can see some sort of leukocyte, some sort of white blood cell. And we know that because we can see a nucleus in here. The nucleus is this darker color, this purple staining body in the cell. And you notice that these cells here don't have any purple staining features. That means they do not have a nucleus, which is what we would expect. All right. So we're going to go on to formed elements of blood. We're talking about platelets. Platelets are important for clotting. They are floating in the plasma. And platelets clump together to form a clot. And what we don't want is them to clot randomly in our blood vessels. So when they're floating in the plasma, they are inactive. And it is only when they detect a breach of a blood vessel or a wound when they become active and start to cling on to collagen and then to each other to create a platelet plug. And then we deposit some fibrin, a protein involved in clotting, which also helps to catch red blood cells. And we form this big clot which closes up the blood vessel, hopefully soon enough that we don't bleed out or exsanguinate. So, platelets are not exactly cells. Instead, they are cell fragments. So we had a hematopoietic stem cell, hema again, and then poietic means that it can become a bunch of different type of cells, so it differentiates into what we need. So they come from a megakaryocyte. So this big cell right here produces the platelets, and then it ruptures and releases all of these platelets, these cell fragments. So again, platelets are a nucleate, and they are quite small compared to the size of the megakaryocyte. Here I just have a little image showing you what would happen if a breach of a blood vessel were to happen, and then platelets arrive to the area, fibrin is deposited, fibrin becomes sort of this strand, this net, which will catch red blood cells and improve the clot, and then our blood vessel will heal. Moving on. So this is going to be a big topic here. We have white blood cells. White blood cells are part of our immune system. So there are many different types of white blood cells that we're going to talk about. They're a large class. So leukocytes, or white blood cells, once again differentiate from a common cell, a hemocytoblast. Not that you need to know that in particular, I just want to show you that all of these different lines come from a very similar place. So over here, if we go back for just a moment, we have our erythrocyte, our red blood cell. And you can see that earlier on, we do indeed have a nucleus. But then, between this step, we eject the nucleus, we get rid of it, and then we wind up with our characteristic red blood cell. Something I want you to note about this diagram, it's nice in that it shows you where this development is happening. So for most of these, the development is happening in the red bone marrow, which is located at the epiphyses of long bones, or the ends of them. Then we have circulating in the blood, so some of these finish out development circulating in the blood, and some of them must go to a certain tissue before they're done. So we have two different types of white blood cells that we're concerned with. There are agranular and there are granular. And that name comes from when you stain them, what can you see in the cytoplasm? So remember, this dark purple staining thing is the nucleus, and then the rest of the cell is the cytoplasm that you can see. Now over here, we have our agranular leukocytes. And you'll notice that the cytoplasm is represented by this translucent bluish color. And this is trying to say that there are no granules in the cytoplasm. Whereas if you look at the neutrophil, basophil, and eosinophil, we have this sort of staining material in the cytoplasm. These are granules. These are things which will be released. Histology. What is histology? Histology is the study of the structure of microscopic tissues. So we're going to look at cells under a microscope and try to describe what they look like, what do they contain. we're going to start off with our list. We're going to start off with monocytes, one of the agranulars. So we have two agranulars that we're worried about, monocytes and lymphocytes. We'll start with monocytes. So these monocytes are suspended in the blood. They float around in the blood, and they are monocytes when they're in the blood. They can migrate to target tissues and then differentiate into macrophages, which you've probably heard of before. So macrophage, you've heard PHAGE, phage. They can do something called phagocytosis, which means that they can engulf. They can surround an invading cell or structure and then dissolve it within themselves. They eat invading bodies. So these are monocytes for you. They represent 2 to 8% of the white blood cell types. So of all of your leukocytes, this is 2 to 8% of the total number. Relatively low amount. Our next agranular leukocyte are lymphocytes, and they are the largest fraction of agranular lymphocytes. What are these? Lymphocytes are an important one for our blood typing exercise we're going to do in a little bit. So we have two different types. We have B and T lymphocytes. And B and T lymphocytes simply describes where they develop. So if you remember this slide right here, we have the T lymphocyte and the B lymphocyte. B lymphocyte is named because it finished maturing in bone marrow. T lymphocytes are named because they left the bone marrow and migrated to the thymus, which is a structure on those sort of surrounding the trachea, very low on the trachea, and they finished maturing in the thymus. So B lymphocytes are important to us because they are the source of antibodies. They are also the source of memory to infections we've had before. So there are B lymphocytes that are B plasma cells. Those are the ones that make a whole bunch of antibodies, which can also be called immunoglobulins, and they secrete them out into the blood. Memory B cells are sort of the storage. They remember infections from the past, so they can become activated, rapidly differentiate into B plasma cells, and then we can have a faster immune response if we become infected with the same thing later on. So what do antibodies do? Antibodies are sort of a tag. So antibodies neutralize foreign particles or target them for destruction. So let's say you have a bacterial cell that's in your blood, so it's humoral. Humoral means fluid, so it is of the blood. So the bacteria is floating in your blood, and it has something on its cell wall or its surface structure, and that thing is an antigen. It can be recognized by a certain antibody that you happen to have in your blood. So eventually those two particles meet and the antibody will bind the antigen, and either these antibodies will completely encase that bacterium and make it inactive because they're blocking a function that it needs, or it can target this bacterial cell for destruction by other immune cells. So T lymphocytes, all that you need to know is that they are responsible for cellular immunity as opposed to where B lymphocytes are responsible for humoral immunity. So the antibody thing is for things floating in the blood. T lymphocytes recognize infected cells of yourself. They recognize self cells. So now let's move on to our granular leukocytes. We have 3 of those that we need to know. Let's talk about the first one, which is going to be the neutrophil. And this is the largest portion of all of the leukocytes, as well as the largest portion of the granular leukocytes. So this, again, is a phagocytotic cell, which means that it will consume foreign debris, foreign bodies, virus particles. And also, because it's granular, it can release antimicrobial effectors. So it can release chemicals or agents that will kill or slow down infections. And we're not going to talk in detail about that at all. But something we do need to talk about is how you can recognize these different types of cells. So once again here I have a histology slide. So we took a blood sample and we stained it, and you can see these cells, and hopefully you can tell me what they are before I say it. These are the red blood cells. I know that because there's a lot of them and they don't have a nucleus or nuclei. They don't have nuclei. And then we have this cell here, which, since it's the only thing different on the page, you can guess is a monocyte. You can see that monocytes are huge compared to red blood cells, and they have a nucleus, and this nucleus takes up most of the cell and is what you might call kidney-shaped. Here I just have a larger image of a monocyte. This is more of the cartoon version, but once again you can see the large kidney-shaped nucleus within the cell. So now we'll go back to lymphocytes. Again, we have these cells here, which hopefully by now you recognize are red blood cells. And then the lymphocytes have a huge nucleus, which takes up pretty much the entire cytoplasm. You can't see any cytoplasm outside of the nucleus. A very large nucleus on these. So now we'll go on to neutrophils. We already talked about their relative abundance being huge and that they release microbial agents. They have a polymorphonuclear nucleus, which just means that it can come in many shapes and it has many lobes. So on your packet it says it has a nucleus with 2 to 5 lobes. So 1, 2, 3, 4, 5. It kind of looks like there's 5 nuclei, but there's not. These are just lobes of the nucleus. And so of this overall image, we can only see 2 neutrophils compared to the dozens of red blood cells within just this little distance. We'll talk about the next relative abundant, eosinophils. These are also granular. So note that in this image we have 2 different types of cells. We have the multi-lobed neutrophil, and then we have the eosinophil, which is specifically a 2-lobed nucleus. And they are attached, though it's hard to see. So if you see a cell that kind of looks like, almost like it's dividing, and it looks like it has 2 nuclei and nothing in between, this is going to be an eosinophil. These are in general a defense against bacteria and parasites. They have a role in allergic reactions and inflammation. So allergies are the result of swelling or edema. What happens is you have inflammation in an area. So something is secreted, usually histamine. Histamine causes your capillaries to become leaky, which means that they let out more fluid than they normally would. And that swelling, that increased blood flow and increased fluid in a certain area is inflammation. Inflammation is increased fluid and heat in a certain area. And so we covered what inflammation is, edema and inflammation. So swelling is not inflammation, but inflammation pretty much always carries swelling. And lastly, of the granular, so we have, remember, 2 agranular being monocytes and lymphocytes. And then we have 3 granular, which are neutrophils, eosinophils, and basophils. These are, again, a defense against parasitic infection and act in allergic reactions, and these release heparin and histamine. Heparin is an agent which prevents blood clotting. So it prevents platelets from compacting onto each other and prevents fibrin from becoming a net and catching red blood cells. And I don't have an image here for basophils, but if we go back up here, you can see that basophils are very dark staining, and sometimes they stain so dark that you can't see the nucleus compared to the cytoplasm. So some more distinguishing factors, eosinophils, aside from having that 2-lobed nucleus, they also have very orangish red granules. So we just covered pretty much all of the formed elements, and where we're going now is talking about blood typing, which I think is more interesting. Particularly the immune system is very important, very interesting, but blood typing is something that we've probably all had experience with, and it's kind of a shame we don't actually get to do this experiment in lab. It sounds kind of fun to do. So we're going to talk about a few things. What we talk about when we talk about blood typing is the ABO blood group. Now, the ABO blood group means that you can have type A blood, type B blood, type O blood, type AB blood, and all of those blood types can be positive or negative. So that positive or negative comes from this thing right here, the Rh factor, which we'll also call D later on. And your blood type is determined based on what antigens are present on your blood cells. So you have red blood cells, and those red blood cells have certain markers. And interestingly, your body does not possess antibodies to those markers. Otherwise, your body would attack your own blood, causing agglutination, which is essentially causing lots of blood clots. Your blood curdles, and it wouldn't flow through your body. You would die. So thankfully, we don't do that. But something that exists that can cause trouble is transfusions. And if you transfuse the wrong blood type, we can cause agglutination. And in fact, that's how we run blood typing tests. So something to note about blood type, some genetics. Remember that you have two sets of chromosomes, right? You have one from your father and one from your mother. And so you have technically two different blood type combinations. So if your mom was type A and your dad was type A, you can be A, but you can also be type O. Because your mother could be type AA or type AO, and your father could be type AA or type AO. If they are both type AO, there's a 25% chance here that you could be type O. Some Punnett squares I'm running in my head there, and some genetics that we're not really going to cover. But keep in mind that these weird combinations that you see, whoa, wait, if father's B and mom is A, how is it possible that we can only get B here when they always be AB? So there's some interesting genetics to consider in that case. And here we haven't considered at all the positive or negative. So Rh factor is something we'll talk about. So we already sort of mentioned this, but we'll mention this in a more clear way. You possess antibodies to the antigens you do not possess, if any. So what do I mean by that? Let's say you're type AA, or just type A. That means you have red blood cells with the type A antigen. You do not have anti-A antibodies, because that would mean your antibodies would attack your cells. But you do have anti-B antibodies, and if you're positive, you do not have anti-RH antibodies. But if you're negative, you do. So that's a little tricky, and we'll talk about that more in a second. But let's think about some interesting cases here. Let's talk about type AB blood. So you have both types of markers on your blood cells that are type A, blood cells that are type B. So you have both markers, which means you neither have anti-A antibodies nor anti-B antibodies. And if you're positive, AB positive, that means you do not have the RH, or you do have the Rh factor, and you do not have the anti-Rh antibodies. And we're actually going to call that, to simplify that Rh factor, we're going to call it the antigen D, because it's shorter, it's easier. So now let's change that a little bit. Let's say you're AB negative. You're still not going to have anti-A or anti-B antibodies, but now you will have anti-D antibodies, because you don't have that Rh factor. Now let's go to the second interesting case of that. Let's talk about somebody who's type O. That means they have an allele from their mom and one from their dad, which means just two copies of the gene which are both type O, so there's nothing to influence it. So if both are type O, and we have a resulting type O blood, that means that we don't have an expression of the A antigen or the B antigen. And let's say that we're O negative. So that means we don't have the Rh factor. We have no D antigen either. That means we have antibodies to everything. We have the anti-A antibody, we have the anti-B antibody, and we have the anti-D antibody. So if you're type O negative, that means you can only receive blood from another O negative. O positive will trigger your anti-D antibodies. A will trigger Maybe the D antibodies also, but definitely the A antibodies. B, again, same thing. So O negative, because it doesn't have any of these antigens on its surface, is the universal donor. So O negative can only receive O negative, but O negative can go to anything else. A positive or A negative, B positive or B negative, AB positive or AB negative. So then, if we go to AB positive, you might imagine that AB positive is the universal acceptor. You can get blood from any other blood type, and that's pretty useful. If you're AB positive, lucky you. If you're O negative, well, then you should be at a blood drive. AB positive. If you're AB positive, that means you have the A antigen, you have the B antigen, and you have the D antigen, which means you do not have antibodies to A, B, or D. So that means if you get blood from type A positive, for example, you will not agglutinate that blood. You do not have antibodies that will attack those blood cells. So that's pretty useful. So donor and receiver information, we were just talking about that. So if you're type AB blood, you have the A and B antigens. And we're not talking about positive or negative here. We don't mention that in this list, but we could do the same. We just have to add an extra one here, an extra one here, here, and here. So if you're type AB, you have the A and B antigens, which means you do not have the A or B antibodies. Who can you give blood to? Well, you can only give blood to somebody who is also AB. But you can receive blood from anybody. Type O, well, who can you give blood to? Anybody. If you're O negative, and if they're negative or positive, that doesn't matter, which means negative or positive is the notation for the Rh factor or the D antigen. So this chart might come in handy for your packet. Hint, hint. Some extra terms. So agglutination, we've talked about this a little bit, and this is sort of the coagulation, the clotting of blood resulting from receiving blood that you have antibodies against. So the coagulation of red blood cells due to the agglutinogen-agglutinin interaction. Agglutinogen is just another term for the antigen. So notice they both end in gen. I just wanted to check that to make sure I didn't give you false information. So agglutinogen is located on the red blood cells. This is the marker of your red blood cells. Agglutinin is the antibody or the immunoglobulin or the agglutinin that can attack foreign red blood cells that have the appropriate marker. So let's say you lose blood and you need a blood transfusion, and they give you blood that's not an appropriate type, and you have antibodies against that. So now your agglutinin will attack the agglutinogen on the surface of the red blood cells, and this is an antibody right here. They are a Y-shaped structure, and you might think that they are going to bind something here in this Y, and that's a logical conclusion. But in fact, they bind at the ends of this Y. So we have two binding sites for an antigen. Because we have two binding sites, that means we can bind two red blood cells with one. So this becomes a linker, right? We kind of stick these to each other. And since we have a whole bunch of antibodies, these two red blood cells are linked by this, these two by this one, and these two by this one. And this can occur over and over again for a huge number of red blood cells until you have a giant clot, a coagulated mess that will not flow through your arteries and veins and can cause heart attack, it can cause stroke, it can cause you to lose circulation in a limb and have to have that limb amputated. So matching blood type for a transfusion is very important. So blood typing in practice, this is the exercise that we are missing from lab. So we have anti-serum, which means that we have a serum containing antibodies of a certain type. And then we can take blood samples. So let's say that we take a blood sample and we're going to put a drop in each of these wells. The control, and in the control we're not going to mix in any antibodies. We would just mix in an equal part of water instead of the antibody serum. So we have water with blood here. Of course, nothing's going to happen. We will stay fluid. Then we have the anti-B serum, the anti-A sera, and the anti-D sera. So notice we have no reaction with the anti-B antibody, which means that we do not have the B antigen on the surface of our blood cells. Over here, we do have a reaction. Notice that the blood is not fluid anymore. Now we have these lumps. We have these coagulated masses of blood within the sample. This is a positive reaction. So the anti-A antibody reacted with the A antigen, or agglutinogen, on the surface of these red blood cells, causing the reaction. So somewhere in this blood, or this blood we can call type A, we have the A antigen. So it's not going to be AB because we already ruled out we don't have any B antigen. We have the A antigen. Now, is it A positive or A negative? You tell me in the next second or two. It is A positive. Here we have the anti-D antibody mixed with the blood, or anti-RH factor. Are we positive or negative for RH factor? We are positive. So these red blood cells have the RH factor, or the D antigen, and that's why we got a positive reaction. So if this were O negative, we wouldn't have had a reaction here, here, or here. We would have all of the wells looking like the control. So I'm thinking that for your exercise this week, I will have a few different cases of this, and you'll have to tell me the blood type that you're looking at. Just like you would have done in lab with your own blood samples. Because, you know, who doesn't miss pricking their fingers with lancets designed for testing blood sugar levels? Sorry you missed out on that. Next thing on your list, we want to talk about the heart. So the heart is super interesting to me, and we're not talking about most of the structures of the heart. We're talking about just some very large, common structures. So the heart is about the size of your fist. It's not as big as you'd think, so that's pretty small to, you know, power your entire body. It is made of cardiac muscle tissue. So remember, you have three types of muscle tissue. You have smooth muscle, skeletal muscle, and cardiac muscle. Well, this is where your cardiac muscle is located. And your heart contains valves which prevent backflow. So your heart pushes blood through your body in a certain pattern, and we don't want the blood to backtrack through that pattern. We want it to maintain that pattern. So we add these valves that only allow the blood to flow in one direction. That is, if the valves are working properly. So let's start off on your list. We'll follow it in order. So there's the right and left atria, or the atrium. So remember that we are looking at the front of the heart as if there were a whole person there in anatomical position. So this over here is the right side, and this over here is the left side. So the right atrium is here. It is fed, meaning blood comes into the right atrium from the superior and inferior vena cava. So blood comes into the heart from the vena cava into the right atrium. And I guess we can talk about the atria separately, and then we'll talk about the order of the flow later. We also have on the left side the same thing, an atrium, the left atrium. So the atria feed blood downwards into the ventricles. So the right atrium pumps blood down into the left, or right ventricle, and the left atrium pumps blood down into the left ventricle. So we just covered the ventricles. We have the right ventricle here, and we have the left ventricle here. We're going to talk about the right and left atrioventricular valves. So atrioventricular, if you break down that word, it kind of looks like it has atrium and ventricle in it, and in fact it does. And so the right atrioventricular valve is over here, and it separates the right atrium from the right ventricle. This valve prevents blood from flowing back up into the right atrium, so blood can only move from the right atrium down into the right ventricle. Same thing with the left atrioventricular valve. Blood can only flow down from the left atrium into the left ventricle. We have semilunar valves then, and we have the aortic semilunar valve and the pulmonary semilunar valve. Semilunar comes from the shape, and the aortic and pulmonary refer to where they're going to. So the ventricles pump blood out of the heart. So remember, blood comes into the heart from the atria and then leaves the heart from the ventricles. So the right ventricle pumps blood out to the lungs, and it does that through this structure here, which we'll name in a bit, the pulmonary trunk. But before the pulmonary trunk, right between the right ventricle and the pulmonary trunk, we have that pulmonary semilunar valve, which prevents blood from going back down into the right ventricle. So it can only leave the heart and go to the lungs, and then in between beats, it can't leak back down. Similarly, on the other side over here, so you see the aorta, this big curved red structure here, red and blue. These are not actual colors. It's just to help describe what, makes it a little bit easier to see the separation. But remember, these are just for animation purposes. So the aorta actually comes down the center of the heart and opens up into the left ventricle. So blood comes into the left ventricle from the left atrium. And then when the left ventricle squeezes, it contracts the blood out. It pushes it up the aorta. And there's a valve between the left ventricle and the aorta called the aortic semilunar valve. Semilunar is shape. Aortic means that the blood is going into the aorta. So papillary muscles and chordate tendineae, we'll have to talk about on the next slide when we see a cutaway of the heart. But I want to talk about some more blood vessel structures here. So the aorta. The aorta is this big structure here. And so it comes up the center of the heart, and it sort of wraps around this right side pulmonary artery. This pulmonary artery, the right side, passes under it. So the aorta is carrying oxygen-rich blood to your body. So this is the initiation of the systemic circuit. So some of the blood will go up into the head, some of the blood will go out to the upper limbs, and some of it will go down to cover the trunk as well as the lower limbs. But it all comes from this, the aorta. So then we talked about where the blood comes back to the heart from the system, from the systemic circulation, and it comes back from the systemic circulation through the superior and inferior vena cava. So you can imagine that the superior vena cava is going to collect blood from the head and upper limbs, and the inferior vena cava is going to collect blood from the trunk as well as the lower limbs. So blood will come back up and then drain into the right atrium. All right, so pulmonary trunk. Pulmonary trunk is this big artery right here. Now, red and blue. Red is usually used to illustrate arteries and blue is used to illustrate veins. However, this is where some confusion comes in. Remember, we said that arteries carry oxygen-rich blood away from the heart and to the tissues, and veins bring oxygen-deprived blood back to the heart so it can go to the lungs and pick up some fresh or some more oxygen. There's one exception to that rule here, and that has to do with the pulmonary arteries and veins. So the pulmonary artery sends blood to the lungs, and the reason it's going to the lungs is to pick up oxygen. So therefore, this artery is still taking blood away from the heart. However, it is taking deoxygenated blood away from the heart to the lungs so it can pick up oxygen. So therefore, the pulmonary veins, normally veins bring deoxygenated blood back to the heart, back to the heart, but these veins are bringing oxygenated blood back to the heart. And we'll talk about this loop more when we talk about the circuits. So before we branch out into the left and the right pulmonary artery, we come up through this big trunk here, which we call the pulmonary trunk. And then we have the left and the right pulmonary arteries. And that's about it for the structures we can see from the outside of the heart. Some discerning features. So notice that the vena cava drains from the back side of the heart, the posterior. We're looking at the anterior. So if you can see the back of this vena cava, then you're looking at the posterior of the heart. Notice that the apex, or the point of the heart, points to the left. It goes into the left side of your thoracic cavity, so over here. Notice that, well, I think those two features should be enough for you to get a view of this. So remember that the vena cava is on the posterior of the heart and the apex points to the left, then you're looking at the anterior. So pulmonary artery is this big guy here, and then the pulmonary veins are these two little ones here, right pulmonary veins and left pulmonary veins. So now we're looking at the interior of the heart. The interior of the heart, we've taken a slice away from it. If you can remember, we've taken a coronal plane. So we've sliced away a coronal plane, and now we can see the interior of the heart. So you can see the valves a little bit better. So let's look at the valves. So here we're looking at the right atrium. You can see where the superior and inferior vena cava are entering. And then right here, if you imagine the front half of the heart were still attached, you would have a closed chamber that would enter the right ventricle through here. And right here is a valve, or specifically the tricuspid valve is another name you can hear for it. But really, it's the right atrioventricular valve. Whatever you remember, tricuspid or right atrioventricular. Now, we have some structures that aid the function of this valve. We have these tendons which come off of the valve. So the valve is pointed downwards. It's kind of like the top of a TP or the top of an arrow. It's a point with three faces. So it's kind of like that from the underside. And it points downwards into the right ventricle. And it opens like this and then closes. So this valve is supported by this tendon, or this group of tendons called the chordae tendineae. And these tendons allow the valve to open like this. But what they do is they prevent the valve from ever prolapsing, or collapsing on itself and inverting. And then you would have no separation between the atrium and the ventricle, and you would probably stop blood flow. Your heart would start fibrillating. It would start like a bag of worms. It wouldn't be able to pump blood anywhere. So it prevents prolapse. And imagine being in the rain on a windy day with your umbrella, and the wind picks up just right and the umbrella flips inside out. That's exactly what the chordae tendineae prevent the valve from doing. Now, the chordae tendineae must be supported by something, some supporting feature, and that's these little rounded, pointed structures that stick off of the heart muscle itself, and these are the papillary muscles. Remember, the papilla means a rounded protuberance, or a rounded sticky outy part. So we have the papillary muscles which attach to the chordae tendineae, which attach to the underside of this tricuspid valve, and they prevent it from prolapsing. We have the same thing for the left atrioventricular valve, sometimes called the bicuspid valve or the mitral valve, whatever name you prefer. And this prevents blood from coming back up from the left ventricle into the left atrium. We want blood to only go left atrium down to left ventricle and then out through the aorta, which you can't really see. It's tucked right up here behind the pulmonary trunk or pulmonary valve right here. So we can see right atrium, right ventricle. We can see left atrium, left ventricle. And we have this big structure in the middle, which is called the interventricular septum, or septum, which most people just call it the septum. Septum is just a wall or a separating feature, just like the septum of your nose. So we have blood come in through the vena cava. It will go from right atrium to right ventricle, and then when the ventricle contracts, it will squeeze the blood out of the left or right ventricle, up the pulmonary trunk, through the pulmonary arteries to the lungs. So we are going to pass through this valve, which is one of the semilunar valves. This is going to be the pulmonary semilunar valve. Remember, semilunar is the shape. Pulmonary is referring to where it's sending the blood. It is sending the blood to the pulmonary trunk. So this will prevent backflow. Now, deoxygenated blood is going out to the lungs. It's going to pick up oxygen and deposit carbon dioxide. and then it is going to come back to the heart through the pulmonary veins. Remember, the pulmonary artery and the pulmonary veins are the one exception where artery and vein, the rule for oxygenation is flipped. So oxygenated blood is coming back through the pulmonary veins, and this is draining into the left atrium. And there are also more inlets for this side over here, which you can't see. It's behind the pulmonary trunk. So blood is draining from the lungs into the left atrium. Then when this contracts, it will squeeze blood down into the left ventricle. And this is the really powerful side of the heart. You see all of this muscle tissue on the left ventricle allows it to force the blood out of the left ventricle, up the aorta, and to all of the tissues of the body, to all of your system, the systemic circuit. So right back here, that would be the aortic semilunar valve, which is going to prevent blood from coming back down the aorta and draining back into the left ventricle. We want blood to move one way and one way only. Here is a superior view of the heart. So we're looking at the heart from the top, and we've sliced off the top half of it. So we've taken a transverse section of the heart. And we're still looking at posterior and anterior and right and left. So still anatomical position, but we're just on top now. So you can see the right atrioventricular valve and the left atrioventricular valve, or the other names, the tricuspid and bicuspid, named for the number of flaps to the valve. And then we have the aortic valve and the pulmonary valve, or the semilunar valves. All right. So now circulation. So circulation, systemic and pulmonary circuits. We've already covered this sort of, but this will be a nice review just to put everything in perspective. The heart is really kind of like two pumps side by side. One is pushing everything through the body, and one is pushing everything through the lungs, and then they're connected right to left by the right ventricle to left atrium. So we have to pick a point to start. Let's start at the right atrium. So systemic circulation refers to the blood that goes to the body and everything else. And then the pulmonary circuits are just that which involve the lungs. So the pulmonary circuit, let's have a look. The pulmonary circuit goes up the pulmonary trunk, out the left and the right pulmonary arteries. And what type of blood is this? This is deoxygenated blood. It's going to the lungs to get oxygen. So now, along the pulmonary circuit, we're going to follow these arteries. And these arteries will get smaller and smaller until we're down to the size of capillaries. Capillaries are tiny structures which connect arteries to veins. And capillaries have different levels of separation so that they can allow cells, well, not cells so much, but fluids and nutrients to diffuse through the walls of the capillary into the tissues, and waste to diffuse from the tissues into the blood and be taken away. So we're going out the pulmonary artery through capillaries of the alveoli. Alveoli are the sacs of the lungs where gas exchange occurs. It's going to pick up oxygen, deposit CO2 in water, and then we are going to work our way back up to the pulmonary veins. Veins bring blood back to the heart. Pulmonary vein will deposit into the left atrium. So that is the pulmonary circuit. Now let's talk about the systemic circuit. We'll start at the left ventricle for the systemic circuit. Left ventricle is the powerhouse of the heart. This is where the really hard squeeze happens. It's going to push blood throughout the body. So when the left ventricle contracts, it's going to push blood through the aorta. And you notice that we've simplified the body for drawing purposes. We have a region that goes up and a region that goes down. So up here would be the head, down here would be the rest of the body. So let's pick the head, for example. So we'll follow the blood up to the head. And so these capillaries don't just occur in the head. There's all sorts of branches which come off that feed into the muscle and the skin and everything of the neck, of the face, and of course of the brain. So we have oxygenated blood coming from the left ventricle that drained into the left ventricle from the left atrium. So left ventricle squeezes, blood goes up the aorta. It's going to deposit oxygen to the cells from the capillaries. It's also going to pick up wastes from cellular respiration. And now it's deprived of oxygen. It's been taken by the cells. There's probably still some oxygen in the blood, but not nearly enough. So we are going to come back from capillaries to veins and work our way back up. In this case, we're in the superior vena cava, and then we're going to drain into the right atrium of the heart. So this is going to be deoxygenated blood. Now this deoxygenated blood is going to drain from the right atrium to the right ventricle, and then we're going to start the pulmonary circuit again. So right atrium, right ventricle, lungs, left atrium, left ventricle, and then the systemic circulation, and then return to the vena cava back to the right atrium. That is our loop. All right, that is all I have for you in this one. I hope you enjoyed the cardiovascular system. It's one of my favorites, one of the most interesting for me personally. Good luck on your practical next week. Let me know how I can help, and be sure to read the instructions for how to complete this week's assignment.